Hi, everybody. Hope you're all doing well. Happy Pi Day, by the way. Maybe you celebrate that. My name is Ian McLaughlin, and I have a PhD in neuroscience from here in Philly, from the University of Pennsylvania. And today I wanted to talk about a kind of study that I haven't really discussed for several weeks at this point. We've been pretty focused on the activities of cells, of genes, diseases, things like that. But today, I want to take a break from that kind of stuff and talk about a study that explores human interaction, and specifically what happens when you trust somebody. So in the past, during live chats, we've discussed how much of our interactions emerge from facial perception, right? Particularly the eyes, the eyebrows, and the mouth. And to give you a sense of what we're talking about here, let's start with what's called the Cooperative Eye Hypothesis. So one of the features of human behavior that's sort of unusual relative to our primate relatives is the degree to which we interact and cooperate in complex organizations. Humans aren't particularly strong, our skin and body, aren't particularly resilient, or, we don't have armor or anything like that. And we're not particularly fast on our feet, relative to potential prey. But there's one thing that we have that our primate relatives and our early, evolutionary competitors didn't have: a particularly versatile brain. And certain features of our anatomies enabled our early ancestors to exploit this unique biology to cooperate in larger and more complex teams to accomplish a whole bunch of different tasks. And so there are quite a few examples of how our anatomies are distinct from our primate relatives, and some are quite a bit more obvious than others, of course. So obviously it's not like, we need to spend time enumerating all of the differences between these two primates, right? A little bit more fur, some different facial morphology, some different musculature. That chimp looks pretty buff, right? But I want you to look more closely than you might have at one particular example of our anatomical distinctions: the eyes. So take a look at this chimp's eyes, and then take a look at the human's eyes. So do you notice anything different? Initially you might have focused on the fact that our human friend down here has blue eyes, while our chimp relative has, brown eyes. And that's true. And it's also indicative of, different kinds of evolution, right? But if that was the first thing you noticed, right, that he has blue eyes, take a second look. Specifically, take a look at the whites of the human eye. So that's a part of the eye called the sclera. And so here are some pictures to make things a little bit clearer. So you're looking at some pictures of chimpanzee eyes and a picture of human eyes. So the first big picture is a typical chimpanzee eye. This chimp has brown eyes, and brown eyes that look sort of similar to mine maybe. But you can also see that just outside of the brown part of the eye, the iris, it's all dark. Chimpanzees don't have white sclera, that we have, that humans have. And as an example, you can see of human eyes in the bottom right corner there. Interestingly, though, every now and then there's a chimp that's born who does have white sclerae. And you can see that on the top right there. So I don't know about you, but at least for me, my reaction to that image of a chimp who has white sclerae is a bit different compared to my reaction to the chimp, or to the other chimp. So it looks like a human is wearing a chimp costume or something like that, like out of Planet of the Apes or something. But so among the various anatomical differences between our primate relatives and ourselves, our white sclerae are central to what I mentioned, the cooperative eye hypothesis, okay, which was proposed in 2002. And so here we go. Hey, Allie. So the concept focuses on the fact that the human eye has the largest ratio of exposed sclerae, right, the whites of our eyes, relative to any other primate. Our eyes are unusually elongated horizontally, in other words, from our nose to our ears. And ultimately, the scientists suggested that these two specific anatomical features of our eyes, that we have these weirdly exposed sclerae, and that we have weirdly wide eyes, were adaptations for enabling our eyes to move even more widely, right, particularly side to side. And that basically enabled us to tell when each other were looking, towards something without having to move our heads, right? And so, in other words, if I move my head to the side, right, I'm looking to the side. But if I'm just tilting my eyes to the side, right, but keeping my head pointed in the same direction, you'll still know that I'm looking to the side by just seeing my face, right? But anyone who can't see my eyes has no idea that my attention has shifted, right? If you're like seeing me from the back, right, and I'm looking over here, somebody from behind me would have no idea. And so this is like almost certainly a super intuitive feature of our consciousness that you likely, take for granted all the time. So, imagine you're sitting at a dinner table or something like that, and you're interacting with somebody across the table without actually saying anything, right? You're just sort of seeing each other. You might express various things by, moving your eyebrows, right? Opening your eyes super widely, like, can you believe Uncle Richard just said that thing, right? Or more importantly, for this whole concept, you need to notice this thing that Uncle Richard is doing over there, right? And so, those types of subtle social interactions, that probably seem completely mundane and almost juvenile, are significantly more difficult for other primates who don't have our unique eye anatomies. And while our primate ancestors weren't necessarily like sitting at dinner tables, of course, amusing each other, with the funny things that our relatives or that their relatives did, right? It almost certainly did help when it came to doing things like hunting prey, or even, interacting with each other among, complex social groups in simpler situations, not at a dinner table, but still, in complex social situations. And so it does seem to be one of those things that's rather intuitive to us, and doesn't have to be like explicitly instructed to kids, right? We just sort of intuitively understand that when I have your attention and we're making eye contact, and if I just gesture somewhere to my eyes alone, you'll know that I mean to direct your attention to the direction I'm signaling, right? By just going like that. And so, to be clear, this is certainly, it's not the only hypothesis to explain why all of our eyes, every human, has a white sclera. And, another suggestion, for example, in addition to the cooperative eye hypothesis, is that it's an indication of how healthy we are, right? Because if we're infected with something, or, one of our multiple organs are failing, our sclerae will begin to change colors, right? From red, if we have some kind of, virus or bacterium that's causing pink eye, or in some cases, COVID-19, we know that has been a symptom in some cases. But also, sclerae can turn yellow if there's something wrong with, one of our organ systems. And so in other words, our sclerae may be one of those things that enables like pretty effective sexual selection, because they're just like a quick sampling of our general health at a time when, we didn't have things like blood tests or medicine or, science, right, in early evolution. So, like basically everything, I'm predisposed to thinking it's a combination of those things, right? It's not like any trait evolves in isolation. But I think the cooperative eye hypothesis is particularly convincing as, at least being a substantial contribution, right, to the white sclerae catching on evolutionarily among Homo sapiens, right? Particularly, and this is a whole, separate topic, but particularly given that humans are unusually good at deception. Being sensitive to something as subtle as the direction of one's gaze as a proxy for, the focus on one's attention can be particularly helpful, right, when navigating sensitive social interactions. And so on top of that, that foundational study, more recent research has shown that human infants tack onto gaze direction, and that, the orientation of the eyes of adults around infants is a very fundamental and critical aspect of the early development of human communication. And so, here's a study that explored this topic from a couple years back, which was published in PNAS, another excellent journal for scientific publications. It's an interesting study, and they review several other studies from a bit back that show and these studies showed things like when someone has an expression with their eyes that looks fearful, there's a greater response from someone, a brain response from someone who's looking at it if the eyes are looking somewhere else, right, rather than directly at the observer, right? And so here, I have an example for you here. And so, it sort of makes sense, right? So once again, you put yourself in the position of a Homo sapien, in a tribe who's like busy, working on hunting and gathering and, defending against potential predators or opponent tribes of primates, or potentially aggressing on, upon other humans, right? If someone makes a fearful face at you, it could very well be because your tribe is invading them. You're the source of fear. Alternatively, if someone within your, and so that's, our Home Alone image is supposed to depict that. Alternatively, if someone within your field of vision is making that face, right, but it's directed somewhere else, you're likely subject to whatever it is that's causing them to be fearful, right? There's an increased reaction when we see someone who's exhibiting fearful eyes that are directed away from us relative to when they're pointed directly at us. And that's measurable within the brain. And so, there's some danger in the environment, right? And it's likely dangerous to all of us. if you see somebody making a face that's fearful, away from you. And to be clear, this is measurable within, the amygdala, for example, which, it's an oversimplification, but it's most certainly involved in the perception and the experience of fear. So infants are like little puddles of chaos when they're born, right? There's a phrase that's used among people who care for infants, the fourth trimester. And astute listeners might recognize that it doesn't make any sense to have a fourth trimester. Right? This phrase refers to the fact that newborns are barely capable of functioning at all after birth, and frankly have no business being outside of the womb, for basically those first 3 months of life. And so, without getting into the details, those first few months can be pretty rough. My daughter, who was born several months before I defended my PhD dissertation, simply would not sleep unless she was somehow connected to one of us, being held by one of us, and preferably my wife, of course. But at least a quarter of my dissertation for my PhD which is several hundred pages, was written with my infant daughter in a carrier of some sort, just resting on my chest, right, while I was sort of like dancing side to side at like 2:30 a.m. Pro tip. If your infant is having difficulty falling asleep and they're in a carrier, if you do a sort of like salsa move, that seemed to work for my daughter. Anyway, so apart from my little trip down memory lane, the point I'm making is that they're really quite minimally functional, these infants, right? But one of the primary things to which infants do pay attention early on is the eyes of the adults around them. And so they will tend to focus on, on the eyes that are directly looking at them, and will fairly rapidly develop a tendency to look at things to which the adults around them are looking, right? Sort of like a baby version of the whole, like, look at what Uncle Richard is doing at the dinner table, type of thing. And so, over the first year of life, the sensitivities of babies improves considerably. And so I remember the first time my daughter, who's now a bit over 2 years old, did the whole look over there thing, which was pretty like amusing coming from a baby. But so interestingly, among young babies who develop autism, this attention towards like the eye directions of the adults around them develops normally for the first few months, but then is actually reduced at around 6 months, making it one of the earlier signs of the likelihood of developing autism or an autism spectrum disorder. And so this whole study basically found that among 7-month-old infants, 7 months, the sclera specifically is critical to how they interpret social cues. Again, the sclera is the white of the eyes. And so they're able to distinguish fearful from non-fearful facial expressions based only on the eyes. And, and it depends quite a bit on the fact that humans have white sclerae. And so the group did, quite a few things, right? It's a complicated study and measured, quite a few things. But, which keep in mind, this is with 7-month-old infants, right? And if you haven't interacted with many humans that are that new, I'll put it this way. Like, I'm always very impressed by studies who manage to collect any data from babies who are that age, let alone data like these, right? With an EEG. So that's what that baby's wearing. So I know that there's pretty much zero possibility that my daughter would tolerate even the notion of putting on a cap like that on her head. We just, got to wear her, I just got her to successfully wear a mask for longer than like 4 femtoseconds, this past weekend. And she's nearly 4 times as old as the babies that are involved in this study. So anyways, so the fundamental thing that this study did with their infants was to measure what are called event-related potentials, right, or ERPs, ERPs, which basically like measurements of the responses of the outermost layers of the brain that occur as a result of some kind of specific stimulus, right? And so, I'll give you an example. I show you a picture of a kangaroo squaring up to box with a human. Weird picture, right? And I've strapped an EEG, on your head, right? And I'm measuring the events that happen in, various areas of your brain while you see this image of a kangaroo and this dude who are about to fight. The changes that occur right after I show you this picture, and immediately, so right as I show you the picture, but then also immediately after I show you the picture, those would be considered event-related potentials, right? The picture is the event, and the potentials are the reactions in your brain that are related to the event. Okay. So they showed these babies some pictures. Let's see. Yeah, here we go. So this is just one part of the study. But so they showed these babies some pictures, right? Some pretty, obviously very simple pictures or figures of eyes that are intended to depict fearful, non-fearful, and averted fearful expressions, right? So fear looking right at you, neutral, and then neutral but directed at the baby, you know, at the observer, and then fearful looking away, right? And so, and importantly, they only showed the images, you know, these images to the babies for 50 milliseconds, right? So 50 milliseconds. That's super duper fast. That's 0.05 seconds. And so that's the kind of fast that you'd likely find to be like imperceptible. Like you might not even realize you saw it, or at least, you know, consciously imperceptible, right? Which is sort of key to the whole story today. But so they showed this picture, or these pictures, to their babies with the intention of showing it below the perceptual threshold, right? So that's why it was just super fast. They did a few other things when performing this experiment, but that was like the main nugget of the experiment. And so you might ask, like, why it was that they went with just like black and white pictures, right? And that's a very good question. And I'm not 100% sure about this, but this is a whole paradigm that's been established, you know, in the field by other scientists. And it includes the eyes from about 50% male, 50% female individuals, right? These are actually based on real pictures of humans. And, you know, they don't describe this in the paper, but the visual perception of babies is still, you know, pretty quite rudimentary. These things will vary between, you know, different infants as they develop. But by 7 months, the baby is likely able to perceive color, to understand, perceive color. And they have, you know, intentional control over their eyes, right? They can move their eyes where they want them to be moved. And since, you know, they're likely at least to be fairly mobile, right? They can, you know, move, crawl, maybe they're starting to walk a little bit, or what is it? Shuffle basically along couches and stuff. Cruise is what they call it. Anyways, you know, so they're fairly mobile, and they have a sense of the relationships between, you know, their body and space as a result, their visual experience, depth, you know, things like that, right? And so, you know, there's no doubt that despite that, there's no doubt that they're still, you know, most sensitive when it comes to like visual perception to high contrast images, right? And so, you know, you might have seen, let's see, boom, boom. Let's do that. You might have seen, here, I can keep this up too. So you might have seen some toys for babies that are colored in black and white, something like this. I think we literally had this specific toy. Somebody gave it to us. And so they look sort of trippy, right? They look kind of strange and weird. But the reason is that there's essentially no greater visual contrast and therefore detectable visual experience than black and white. Right? And particularly in the early days of visual development, babies can really only detect and perceive super high contrast visual experiences, right? So that's why you see so many black and white toys. And I suspect that's probably why they went with this paradigm has been established using black and white eyes. And so they're measuring activity primarily in the occipital lobe. Oh shoot, I meant to include a schematic. Maybe I'll show it at the end. The occipital, it's just in the back. Oh, you know what? I have this brain in a jar. I forgot I had that. So the occipital lobe will be sort of like back here. This is not a real human brain, if you can't tell. Anyways, so the back of the brain, right, the occipital lobe, which is basically, you know, this is where the visual cortex is. It's a part of the brain that processes our visual information. And it's a huge proportion of our brain that's devoted entirely to various aspects of vision, color, movement, these types of things. And so the group found that even at 7 months old, infants detect social cues from the sclera specifically, right? That's the white thing that you're seeing in these images, the sclera. And so, you know, even at this young age, infants could distinguish between fearful and non-fearful expressions, as well as direct and averted gazes from just 50 milliseconds of a black and white image of basically just the sclera, right? So this is about as hardwired as a social reaction can get, right? No, you know, obviously nobody sat them down and had them study images of different people to memorize what fearful and, you know, neutral eye expressions look like, or, you know, directed versus averted gaze, right? It's not like they studied this, right? And so, or it's not like the infants studied this, right? And so obviously, you know, this team characterized, you know, more of like how the brains of these infants processed these perceptions. But I'm highlighting this study just to drive home that the fact that facial perception, and eye perception in particular, specifically of the sclera, is fairly central to, you know, how it is that humans interact with one another, particularly when not even speaking, right? And so I know that was a sort of long wind up to the actual study that I wanted to talk about today, which came out a little while ago, published in Scientific Reports, which is another highly regarded journal for scientific publications. And so, okay, so we just talked about how much attention we devote to something as minuscule as the whites of our eyes, right? Something is starting at a very, incredibly young age, at least by 7 months, if not earlier. And so this study explores some of the other interesting aspects of social interaction. Weird sounds. Okay, of social interaction. And aspects of social interaction of which you're almost certainly completely unaware. And so this is a team at Leiden University in the Netherlands. And fundamentally, what the team shows in this study is that our brains and bodies interact with each other in ways that we don't necessarily see or hear, and barely even feel, probably, at least not consciously. So we're talking about things like heart rate, skin conductances, jeez, and another part of our brain, another part of our eyes can begin to become synchronized, right? The pupil. So they call it autonomic mimicry, okay? And so this team worked with 152 participants, and they were playing a game that's frequently used in psychological as well as philosophical experiments, in thought experiments, a game called The Prisoner's Dilemma. Okay, and so the prisoner's dilemma is like one of those things that comes in a variety of flavors, right, different iterations, but it's basically used to probe things like game theory, but also morality, trust, right, some of the most complicated aspects of how humans interact with one another. And so here's the basic version of the prisoner's dilemma. Okay, 2 members of a criminal organization of some sort are apprehended by the cops after committing some kind of crime, something like robbing a bank or something, right? Each one is immediately separated and isolated from each other, right? They can't talk to each other, and are completely incapable of communicating, right? They're in social isolation in totally different cells. Can't hear each other or anything. No communication. So despite having these criminals in custody, the prosecutors, which is basically what we call lawyers who are like working to prove the guilt of the prisoners in the United States, they don't have enough evidence to successfully convict both of them of the whole crime. Okay, so they need more evidence to convict the prisoners. So the prosecutors come to the prisoners with a bargain. Okay, they provide each prisoner with a choice. The choice before each prisoner, is to either betray the other one by testifying that the other prisoner committed the crime of robbing the bank, or of course each prisoner can refuse to cooperate and remain completely silent and not cooperate with the prosecutors or the cops. So to summarize, right, the incentives in front of each prisoner are the following. If both of the prisoners snitch on each other, both of them will serve 2 years in prison. If one of the prisoners snitches on the other, but the other stays silent and refuses to cooperate, the one who snitched on the other will be set free, and the other will serve even more than 2 years in prison. However, if they both remain silent and refuse to cooperate, both of them will only serve a single year in prison, because the prosecutors will fail to successfully demonstrate, the comprehensive guilt, right, for committing the crime. And so, this is obviously like an artificial situation. We're not to concern ourselves with like what happens if one of them is set free, if there's any kind of retaliation down the road. Don't worry about any of that stuff. The whole purpose of the prisoner's dilemma is to assess that single choice in that moment when the prosecutor comes to each prisoner with, this choice of either betraying their partner in crime or remaining silent, despite the knowledge that they will have to serve a year in prison, okay? Again, there's a ton of different iterations of the prisoner's dilemma that can help explore the contours of how these decisions are made. Things like increasing the prison time if they refuse to cooperate, or increasing the prison time that the other prisoner will have to serve if one of them snitches on the other, right? But that's the fundamental premise, okay? So that's what they used in this study at Leiden University. So they did a few interesting things in this study. But they had 152 people play a slightly modified version of the prisoner's dilemma. And so, these 152 people played it 30 times consecutively. Interestingly enough, they were facing each other, right? They could see each other in these 30 trials, but they weren't able to talk to each other, right? They could see each other's faces, but no verbal communication. And then in addition to those 30 trials, where they could see each other, they did another 30 consecutive trials, separate trials, of their version, right, of the scientist version of the prisoner's dilemma without being able to see each other. Okay, so here's how that looked. So you can see here, this is the situation where they can't see each other, and they're playing the prisoner's dilemma, and here's the one where they can see each other. And I know you might be thinking this is sort of like a low-tech way to manage whether they could see each other or not, but I think it's quite good, actually, that they didn't do it via like computer screens or something like that. People were either literally seeing each other, seeing their faces, their facial expressions, the movements of their eyes, these types of things, or they were looking at a blank wall. Okay. There are all kinds of nuances that can be missed, particularly, there's all kinds of nuances that are missed when they're like literally mediated, right? When seeing someone's face via video. And apart from all the headgear, right, that these ladies are wearing, they were straight up looking at each other. Okay. So the main difference is whether or not the people playing the prisoner's dilemma could see each other. Okay. So, what makes this an interesting study is that they collected data to measure what was going on in the body while these participants were playing the game. And so they pretty much measured 4 main things: heart rate, skin conductance, and skin conductance is basically like changes in sweat and blood flow in the skin, moment to moment. Okay. They also measured, things like the muscles that enable us to smile, around the mouth, eye movements, things like that. That's what they were measuring. And there's a kind of theme to what they were measuring. These are things that are almost entirely controlled unconsciously. They're responses in the body that you can't really control, with like conscious effort. They just manifest as a function of your inner reactions, right? So you might have heard that when somebody's around a person that they find attractive, their pupils will tend to dilate, right? It's not, and that's an example of these unconscious things, right? It's not like a person decides like, now is the time for me to dilate my pupils because I find this person attractive, right? Obviously, it just happens. And there's a sort of, by the way, this is a kind of an interesting, there's a kind of like a reciprocal effect here. So men will tend to rate pictures of women with pupils that are dilated as more attractive compared to those without dilated pupils. And to be clear, I mean that like a picture of the same woman will tend to be rated as more attractive if their pupils are artificially dilated relative to one where her pupils are smaller. Bigger pupils, more attractive, right? And so, by the way, and I know I'm kind of indulging myself here, but stick with me. I think this is an interesting story. So dilated pupils were known to be attractive many, many years ago. This isn't a new thing. In fact, atropine, the substance that your eye doctor, uses to dilate your pupils, or probably, would have used before we had newer drugs that could do the same thing. Atropine is derived from a specific plant. So the plant, oh shoot, let's see. Okay. So yeah, from a specific plant. Sorry. And this plant, the name for this plant is Atropa Belladonna. Okay. So, and that's this plant up here. So it's not the only plant that produces these compounds. In fact, one a particular plant is quite popular throughout a few neighborhoods that I've seen in South Florida. So here, let's see if this one works. Here we go. Yeah, in South Florida, right there. So it's a little surreal that the plant is so prevalent, despite, it's a very beautiful plant. But that also produces atropine along with other similar compounds that could be rather dangerous. I won't get into the details because, it would be bad if people just started going out and exploring. It can be rather dangerous. Anyway, so the name belladonna, right, is evidently derived from Italy, where it was evidently used to dilate the eyes of women to achieve, an intended attractive effect to the eyes of men, right, during the Renaissance era, like the 1400s, 1500s. Bella, beautiful, donna, woman. Pretty cool, right? A little bit disturbing maybe too, but anyway, so back to the study by the group at Leiden University. So the group found that physiological synchrony, okay, was associated with cooperative success in the prisoner's dilemma. And particularly when this was true, particularly when, the participants could actually see each other when they were looking at each other's faces. It was specifically skin conductance that seemed to synchronize and not so much heart rate. And so, when they say cooperation, they're referring to the outcomes of the prisoner's dilemma game, right? And so, like we discussed, if one of the prisoners decides to cooperate, there's a risk that they will, be exploited by the other prisoner if they decide against cooperating. And so, if the other person decides to snitch on you, you're going to prison for quite a while. But if neither person snitches on the other, both suffer a shorter time in prison, and you have no way to know whether or not the other person is going to snitch, right? And so it turned out that when people who were playing the prisoner's dilemma game, they tended to synchronize. Autonomically, that is. Components of our biology appear to get in sync when we're making decisions that hinge on trust. And so if our skin conductance is similar, it seems like we're on the same page, and we'll cooperate in a way that produces better results for both of us than if one of us betrays the other. So what do we learn from this study? Well, and they did quite a bit more than just that specific experiment, but I think that was the sort of the main event. So what do we learn from this study? Well, it can at least help us to answer one specific question. How can we tell if we can trust somebody? Or more accurately, how is it that we end up trusting somebody? Obviously, it's more complicated than the results from this study, right? That's just one measure. Quite a bit of the inner deliberation is obviously subconscious. While the experience of deciding, if you want to call it that, to trust somebody is consciously experienced, right? You feel like you have decided to trust somebody. But part of that deliberation results from what appear to be literally synchronization of certain physiological events that just happen to us, right? They just emerge. We have no control over them. They're totally subconscious. And so it seems to be the case that like literally seeing each other's faces plays a meaningful role in the emergence of trust between people, right? And I suspect this is pretty intuitive to most people, right? When you're like literally hanging out with someone, not on like FaceTime or Zoom or video chat of some sort, there's just a lot more substance to your interactions compared to like a video chat or a phone call, or certainly text messages, right? When it's just written word. So I inevitably I have to wonder what the outcomes of this study would have been had they conducted their experiments with an additional group that use something like a video conference platform, something like Zoom or Google Meet, right? Given that, a lot of the world has had to adapt to current circumstances by using these platforms, it'd be very interesting to see if any of these kinds of subconscious interactions happen at all during these mediated interactions via, video chat. And so, ultimately, I see this study, and I find it interesting, because I think it adds to a sort of a wealth of evidence that suggests that our interactions involve much more than just the words we say to each other, right? Important as those words are, of course, the subtle things we do and that our bodies do, particularly the subtle things over which we have no conscious control. They seem to play pretty substantial roles in determining how we ultimately interact with each other. And those subtle things emerge as early as when we are only 7 months old. So pretty hardwired. All right, that's what I have for you today. Thank you all for hanging out. Oh, cool. Yo, folks, smash that like button. Thank you so much, Cosmic Rose. Hey, what's up, Watchmaker? Good to see you. Home Alone, yeah. Fascinating. Just can't type fast enough to comment right away. I have a concussion, and I'm on limited screen time, so I'm sorry. I'm listening extremely interested. Oh, geez, I'm sorry to hear you had a concussion. That is no joke. Hey, Scott, good to see you, man. Hopefully it wasn't too severe. Concussions are like one of those things that I think are frequently underestimated. But there's a wide variety, right, in severity and how people recover from them. So best of luck there. The scared face sympathetic reaction was discussed in the making of the documentary in the American remake of The Grudge. Is that right? That's very interesting. That was a pretty good movie. I mean, it was a horror movie, but I thought it was pretty good. The bounce and rock was it for my son. Yeah, my sister used a yoga ball, which was smart because, she would sit on the yoga ball, but just because you get tired, it's like quite a workout. I was literally standing for like 7 hours sometimes and dancing basically. Like, it's no joke. Kept me in shape, my daughter. So don't make fun of the guy getting beat by the kangaroo. My cats kick my ass all the time. Where does trust come in? Yeah, okay. So I know it took me a while to get there, but that was the whole purpose of the prisoner's dilemma experiment was, do you trust the other person that you're literally seeing? Do you trust that they are going to make the choice that is better for both of you, but that depends upon you making that same choice? Do you trust them to make that choice? And the point is that, as we come to the decision of either trusting or not trusting somebody, there's a lot going on subconsciously and autonomically, physiologically, that seem to either need to happen or inevitably happen when we trust somebody else. So we can get rid of where? Oh, right. Okay. Jeez, I have so many figures in this one. Okay. Thanks for bearing with me. Also works across species. I faked out dogs and cats by looking at a point behind. Yes, that is an excellent point. I actually meant to bring that up. Thank you, Scott. Yeah. Man. That, I can't believe I forgot to do that. Because, you know, not, as I was saying, like, essentially no other primates have white sclerae, right? Whites of their eyes. Dogs do. Now, they're not as big as humans, but they do have a white sclera. And, you know, of course, I don't know if there's any greater symbiosis between, like, two large mammals than dogs and humans. And yeah, I mean, that is one of those things. And again, you know, very, very small, but you can tell, right? Or you can tell when they're looking around. And they can tell, yeah, and they are sensitive to it as well. They're sensitive to the cooperative eye hypothesis. Really frustrated I didn't bring that up. Thanks, man. Okay, this is fascinating. I'm listening and have so much to say and ask, but I'm healing from a concussion. Yeah, no, take your time, please. I guess that explains why whenever I'm talking to a woman, her pupils are pinpoint tiny. Yeah. All right, all over my neighborhood. Oh yeah, datura, right? Yeah. And, you know, every now and then, it's not super common, but every now and then you'll hear a story of, you know, somebody reacting pretty badly to datura. Yeah, I remember in, like, the earlier days of the internet, people would post stories of, like, them intentionally ingesting the substance. Crazy stories. Yeah. So, okay. Oh, yeah, what about trust while texting someone when you cannot see or, you know, exactly. See the other person's face. Exactly. I mean, there's so much to primate interaction, human interaction, that, you know, I just don't think that we appreciate. But I think we intuitively appreciate, right, that things like, you know, if I'm, like, gesturing over there and only one person can see my eyes because they're sitting across from me at the dinner table or something, right? I mean, you know, I did that as a kid for sure. And I certainly had no idea that I was, like, exploiting this unique anatomy that evolved in humans, you know, uniquely, that enabled us to cooperate. And in that case, it enabled me to cooperate with a compatriot of some sort without my parents knowing. Okay, why don't we start the live hack? Thanks for watching, everybody. By the way, if you enjoy these topics and you haven't subscribed yet, perhaps you'll consider it. And yeah, I appreciate it. Okay, here, let me do this. Hello, everybody. My name is Ian McLaughlin, and I have a PhD in neuroscience from the University of Pennsylvania. And just now on YouTube, youtube.com/anthropoid, I talked about some interesting studies that can help to explain what happens when you trust somebody, when you develop trust, right? When you, let's say, decide to trust somebody. And so I went on a bit of a roller coaster through, like, you know, historical hypotheses and, you know, just a variety of studies. But I think it's an interesting story. And also the cooperative eye hypothesis, right? The fact that humans have white sclera, the, you know, whites of our eyes. We're essentially the only primate that has that. Every now and then there's another primate, you know, from a different species that's born with white sclera, with whites of their eyes. But they're very, very, that's very, very rare. And all humans have white sclerae. And so there's a bunch of theories as to why it is we have white sclerae, and the evolutionary consequences of that. Oh, every single time. Okay, let's just do this. All right, is substance dualism valid? Man, I love it. So, right out the gate with, like, hardcore philosophy. So, substance dualism is just, so if anybody's unfamiliar, substance dualism is essentially, it argues that it's a model to understand human consciousness, and it essentially argues that consciousness emerges from more than just material, right? More than just the physical wet work of the brain. Dualism was, and so, you know, fundamentally, substance dualism argues that there are two types of substances. There is, like, the stuff of biology, and then there's the stuff of mind. Okay, and so, and these are, you know, distinct components that generate consciousness. Dualism was probably the dominant theory to understand consciousness for a very long time. And, you know, there are certainly some people today who still sort of adhere to a kind of a version of dualism. You know, I think the most justified would be compatibilism, and which is essentially like, you know, it's not as strong of a position on substance dualism. So basically it's like, yes, the brain is very, very important to generating consciousness, and without a brain you won't have consciousness, but consciousness emerges from more than just the brain. And so, okay, so but the initial topic was, is it valid? Well, I can only tell you my respond from my personal experiences in the lab, the experiments I've conducted, obviously the classes I took, and the other neuroscientists with whom I interacted. I don't think I've ever met in neuroscience a dualist, like a strong dualist. I think it's safe to say that today most of the field is no longer dualist. And so when you say, so why? Like why don't I think that substance dualism is valid? So, there's a bunch of reasons. But I think, dualism arose in an era where, we didn't really have such a thing as neuroscience, certainly not in its modern incarnation. And we just didn't have the tools to interrogate the brain on the levels that are the most important to how it regulates consciousness. Now we do, and we have had for decades, arguably longer. And so, there's a variety of ways to sort of highlight these data, but like fundamentally, one sort of maybe perhaps intuitive way is, if I remove, let's say, your thumb, you'd probably be pretty upset with me. It would change, how you interact with the world. You would, it would be unpleasant, I'm sure. Having opposable thumbs is quite handy. But nothing fundamental to who you are, to your consciousness, will change, right? You'll still be the same person, just a little bit angrier. However, if I remove a fraction of the size of your thumb from the brain, you will be a fundamentally different person. In some way, and depending upon what part of the brain is removed, that will determine which parts of consciousness have been, changed forever. And so, it's evidence like that. This stretches way back. So, you might have heard, or you might have seen probably, some of the really beautiful anatomical drawings from, hundreds of years ago by a guy named Andreas Vesalius. If you saw a picture, he was essentially an artist and an anatomist at a time, by the way, when it was essentially verboten. It was like literally sacrilege to do the kinds of experiments he did. And so, the kinds of anatomical, explorations that he did. And, this is from a couple hundred years ago. And up until Vesalius, the only anatomical, knowledge that was available was from like ancient Greece, from Galen. Okay, so, Andreas Vesalius collaborated with, at a certain point, and I'll keep this story as short as I can, there was a jousting tournament, and some monarch of some sort participated in the jousting tournament. And, jousting is when people are on horses, they have those big poles, and they ride at each other and then try and skewer each other. And so this monarch was knocked from his horse, and it turned out that some wood impaled the back of his head. Not great, particularly at that time, particularly at that level of medical sophistication. But so the two most prominent anatomists in the era were summoned. Andreas Vesalius and then a guy from France named Ambroise Paré. And they began to notice things about how the king was changing. Most notably, his vision was essentially obliterated. And now we understand quite a bit more about the brain. And so here I have this brain in a jar. Right about back here is where the visual cortex is. The visual cortex in the occipital lobe, that's right at the back of the brain, right where the king happened to have been impaled with some wooden spikes. And so then we learned, it's this part of the brain that is critical to vision, to visual perception. And, that's just an early version, but, basically the story of early neuroscience was learning some of the different functions of the brain by observing what happens when some specific part of the brain is destroyed or is somehow aberrant. A very classic example would be HM. So HM, Henry Mollison, he was a guy who had very severe epilepsy, and the only solution at the time was to basically remove his hippocampus. It's not, I don't think it was the entire thing, but it was a large portion of his brain, totally removed. And thanks to HM, we learned that it's this specific part of the brain that's critical to specific, components of memory, right? He could still function. Probably if you were talking to him, you would notice maybe something was a little up because his conversation would be kind of maybe staccato, meaning like, he might forget what topic you were talking about, these types of things, but he could function, he could feed himself, clothe himself, and so on. And so, this specific part of the brain controls or regulates this or produces this specific part of consciousness. And so the story of early neuroscience is a sort of progression of those types of observations. And so essentially over time, the amount of unexplainable aspects, or the number of unexplainable aspects of consciousness has shrunk basically to the point where I think the burden is on the dualist. It's no longer on the materialist or the physicalist or the identity theory. Identity theory has nothing to do with like personal identity. The let's say hardcore neuroscientist who's arguing no, the brain is sufficient. The brain produces consciousness. Used to be the burden would be on us to demonstrate sufficient evidence to be convincing to convince a dualist. At this point, I think the dualist is the burden of evidence is on them, because for what reason do we need to invoke some other, form of some other substance to explain consciousness? So that would be, I could go on and on. I know I've already gone on, but, at heart, I'm really a neuropharmacologist. That's how that was my gateway. That was my gateway drug into neuroscience. Just the fact that you can take just one tiny little molecule, let's say ethanol, right? Widely consumed drug. It's literally just 2 carbons and a couple hydrogens. That's it. It's a tiny little molecule. Just one little molecule. When it gets into the brain, fundamentally alters your consciousness. Potentially, quite drastically, such that you either pass out or you become, giddy or, whatever, gregarious or aggressive or whatever it is. Just that one molecule. And it's not like that molecule is causing these changes in behavior because it somehow has like a metaphysical property that enables it to interact with, a non-physical, substance. No, it's because it changed the way the brain is the activity in the brain. That's what produces drunkenness, all right, or being tipsy. And so, on and on we go. But so, yeah, for me, I think, there's just so many lines of evidence that have demonstrated that we don't need something else to explain consciousness. It's remarkable, it's magical, it's amazing that just this 3-pound mass of about 86 billion neurons, about as many glial cells, a lot of fat, a lot of water, a couple proteins, that's it. And, with the right connectivity, with the right architecture, you get something as versatile, as comprehensive, as effective as human consciousness. Okay. Sorry I went on for so long. Oh, thank you for the super heart, Victoria. I appreciate that. By the way, I should note, so I have a YouTube channel, youtube.com/anthropoid. I just talked about some of the very fundamental changes in your body and in your subconscious that occur when you end up trusting somebody. And I also talked about a whole lot of other stuff, but you can check it out. I have a link to that in my profile page here on Periscope. But I also, given that Periscope will be shuttered in a few weeks at this point by Twitter, I have been using a new live streaming platform called HAPS, HAPPS.TV. It's a great platform. The community is growing. They have a lot of tools that enable me to do a lot more than just speak. And so I have a link to that in my profile as well on Periscope. Go check it out. And also, I have to say, I've seen a lot of platforms come and go over the years, over these past, 5 years. And there are some specific features of this platform, and specifically the founders and the management team, that are really distinct from any other social media platform in general, let alone live streaming platform. So check it out. I think you'll dig it. Haps.tv. Okay. Do you think weed can save us from coronavirus? Do I think weed can save us? No. No, and listen, this is coming from somebody who totally advocates for the complete legalization of cannabis, recreational cannabis, as was just done not too long ago in the state of New Jersey, joining the ranks of states who have seen the light and have made the only ethical decision to do so. But regardless, cannabis is not a panacea. Maybe it might help, with some of the unpleasant after effects of COVID-19. But I suspect, particularly for like long haulers, but I suspect for a respiratory disease, COVID-19, that can just ravage the lungs, probably the last thing you want to do is add some combustion byproducts, right, to burned matter into your lungs. And I know I'm completely aware of the literature that indicates that cannabis does not present the same level of carcinogenicity, or it doesn't necessarily cause cancer, the smoke of cannabis, to the same extent as tobacco. I know, but listen, if you have COVID-19 and your lungs you're literally struggling to siphon oxygen from the breaths you're taking because, there's so much inflammation in your lungs, probably cannabis isn't going to be your top priority. But hey, edible cannabinoids, particularly for people who have, chronic pain. In some cases it can exacerbate chronic pain. Obviously, chemotherapy for the treatment of cancer, a variety of maladies, or just because it's fun. People enjoy it. Edible cannabinoids are always an option. And so certainly if I tested positive for COVID-19, I would probably, and I smoked weed, I would probably transition to edibles if possible. Okay. He had skeletons in his closet. I'm not sure what you mean by that. Oh, like the gut. Is the gut-brain connection real? Oh, yeah, Scott, for sure. Yeah. So Scott is referring to something called the enteric nervous system. So, there's a lot of exaggeration that takes place when it comes to the gut-brain. But it is a remarkable feature of our biology. So the exaggeration comes in. Well, let's just talk about what it is. So we have a ton of neurons throughout, outside of our brain, right? Just a huge number, and many of them reside within the gut. And the gut nervous system is sophisticated to the extent that it can actually function completely independently of the central nervous system, right? It'll do what it's got to do independently of any input from the brain. So it's kind of a rare feature of our nervous system. The gut nervous system produces a full complement of neurotransmitters, serotonin, dopamine, etc. It has a ton of different types of neurotransmitter receptors. So like on paper, it's very similar to the actual brain. On paper, if you're just listing the criteria, right, it has a ton of genetic expression, many proteins are expressed, there's neurotransmitters that are released, there are neurotransmitter receptors, there's tons and tons of different types of neurons. They interact with stimuli, they react to stimuli. However, and so that's on paper, they look very similar, but however, the main difference, the secret sauce of the human brain is the connectivity of the human brain, right? We have 86 billion neurons, about as many glial cells, things like astrocytes, oligodendrocytes, microglia, just other types of brain cells that they don't get as much attention as neurons, and they're probably not quite as important as neurons. But anyways, we have billions and billions of these neurons, and we have trillions and trillions of connections between them. But if you just had a bucket of neurons, right, and they were just neurons, they weren't arranged in any kind of particular coherent way, certainly not into a brain. If you just had a bucket of neurons, that bucket is not going to be sentient, right? Just a ton of neurons does not make for a brain, or certainly consciousness. And that's sort of what the gut nervous system is. It's a ton of neurons that executes its roles by physiological role very effectively. Don't get me wrong, but that's what it does. It does not produce consciousness. And the same goes for, a lot of people are very interested in things like the vagus nerve. The vagus nerves is probably a more anatomically accurate way to depict it. And the nervous system around the heart, people are very interested in that. But same thing goes, right? Just because you have a bunch of neurons in an area performing a function very effectively, like the neurons around the heart or the gut nervous system, that doesn't mean that there's sentience or that there's consciousness. It's the connectivity between the 86 billion neurons that we have in the brain that generates consciousness. So if you believe substance dualism is invalid, the issue of free will emerges. Are you truly free thinking? Justin, Just Witten. Yes, I mean, you're 100%. I couldn't agree more. That free will is, or the conversation about free will is inextricably linked to sorry, I'm trying to get, okay. Free will is inextricably linked to the conversation about of dualism, right? Because so what I'm going to say, maybe your name is Justin. So what Justin is arguing is that if consciousness emerges, or what, I don't know what he's implying, I don't know if he believes this, but if consciousness emerges exclusively from the physical stuff of the brain, right? There's no other matter, no other substance that generates consciousness. It's just the brain. And that physical stuff obeys the laws of physics, right? It's not like there's some kind of magical property to the brain. It obeys the laws of physics just as much as this pen does, right? It's just more versatile. Then that would mean theoretically that every interaction that occurs in the brain would be predictable, right? Right? We can predict where, Venus is going to be in the solar system, a trillion years from, well, maybe not a trillion, but, hundreds and hundreds of billions of years. I don't know why, maybe not a trillion. Well, I guess maybe it'll be eaten up by the sun. Whatever. Let's go with a billion years. We can predict pretty precisely where that planet is going to be, right? So similar dynamics at play within the brain, right? It's just that there's so many domains of interactions and you would have to predict where each atom would be, let alone each neurotransmitter, each neuron itself, each synaptic connection, the activities of all these neurons to be able to predict that kind of stuff. But theoretically, you would be able to. So the question emerges: is free will? Does it exist? And so that's really the genesis of what I refer to compatibilism. It really is generated via a concern for free will for people who argue, neuroscientists and neurophilosophers who argue that it does indeed exist. But for me, and so you see the issue with free will in the genesis of compatibilism. If free will exists, then having a deterministic biology, having a brain, just the physics within the brain playing out, generating consciousness, then you need something else for there to be free will, right? Some of the reflexes that people have to justify or protect free will is, well, humans, perhaps that's true. Perhaps, human consciousness is a byproduct of deterministic phenomena, but perhaps there's some randomness to it. Perhaps it's not predictable. And so free will emerges from just the fact that you never know what's going to happen. You never know if a neurotransmitter receptor is going to be, in some cases, going to be released or not released. So for me, that is not free will. Just something that's just random doesn't have free will, right? And so yeah, I mean, if you were to ask me, so my PhD was devoted to understanding the neuroanatomy that underlies anxiety and addiction. And the reason I think addiction is both an important thing to study, just because of the degree of human suffering that emerges as a result of addiction, particularly in the United States, particularly right now, more people have been lost to opioid overdose fatalities or just drug overdose fatalities than ever in the history of the country. So it's important just for that, just for the medical, it's just a critical public health concern. But beyond that, I think it is an ideal venue to explore human behavior, human decision making, and free will, therefore, right? Because, talk to 9 out of 10 people who are addicted to heroin. They don't want to be addicted to heroin. The same goes for cigarette smokers, right? I think I've never met somebody who smokes cigarettes who's just like, this is awesome. Like, I just love it. I'm not trying to quit. I never tried to quit. I'm not going to try and quit. Right? I've literally, I think without exception, everybody who I've met who smokes regularly just says, I hate it. Like, I hate the smell. I hate that it makes me smell. I hate that other people hate how I smell. And, I would prefer to quit, but it's just so difficult, right? So where's the free will in that? And so, yeah, that's why I think addiction is just such a great. In neuroscience, we learn about human consciousness by perturbing the system. That's one of the major strategies, right? Like the king falling and impaling the back of his head, having his vision destroyed. That perturbed the system. But also you can perturb the system with pharmacology, with adding in a little bit of drugs, or adding in some electrical current to specific parts of the brain, or infecting a certain part of the brain with a virus, seeing how it changes the output of consciousness. And so I think addiction is an ideal phenomenon that occurs as a byproduct of human consciousness that helps us to understand free will. And so if you're asking me if I think free will exists, unless you redefine free will, which is another move that my favorite philosopher who's living, she does this, Patricia Churchland. She argues, okay, maybe the popular conception, if you were to walk up to 10 people, on Broad Street here in Philly and ask, hey, what do you think free will means? It's one of those words that I think we all sort of understand what each other means, but I think, you'll get like 10 different versions of free will, right? Some of them will of course be, religiously inspired, but not necessarily. And so, she argues that, okay, maybe free will, in the sort of the strongest version of free will, where we have our behavior, our consciousness is not limited by anything. Every decision is made, unfettered. It is made free of any kind of external limitation. Okay, maybe that doesn't exist. But maybe what we should do is, first of all, not concern ourselves with free will. That's really what she argues. But let's just redefine free will as the thing that humans do, right? Or the things that humans do, right? That if a human is capable of doing it, that's what we're going to call free will. Okay, that's different. Then sure, free will exists. But then for me, it's just a semantic difference, and it becomes a sort of a pointless term, right? Why aren't we just calling it consciousness then? So if you were to ask me, free will as it's widely appreciated as meaning does not exist. At least I have never been able to find an example that would, open up any space within our nervous system, within our neurobiology for free will to exist. And that has a bunch of very important consequences for society. Probably most notably, immediately criminal justice. Right, so let's just assume, and I think actually when it comes to free will, while I think dualists are certainly the minority in neuroscience, I think free will is a little bit different. There are definitely neuroscientists who are very strong advocates of free will, who believe it exists, and so it's not just like 9 to 1, right? When it comes to like dualism, it's probably a little bit more than half and half. Though in my experience, more neuroscientists with whom I've interacted do not believe free will exists than do. But definitely some do. Some very prominent neurophilosophers do. I think Daniel Dennett still argues that free will exists. He's a compatibilist. And as I said, Patricia Churchland has a sort of a unique take on it. But what she really argues is we should think about culpability, right? We should think about the consequences of human behavior rather than just like, ruminating over whether or not free will exists. But, when it comes to criminal justice, if free will doesn't exist, then what is the point of punishment, right? Or I should say, what's the goal of punishment, right? In the United States, but I think throughout, most of the world, our criminal justice system is basically based on retribution, where you did something wrong, you deserve the punishment you're getting, right? You earned this. It is justice for you to be punished for this thing that you did, rather than, an alternative framework would be, we use criminal justice to mitigate harm to society, right? Where if somebody commits some kind of heinous crime, you're not punishing them because they deserve it, right? Because again, if free will doesn't exist, what do we even mean by they deserve it, right? They had no control over it anyways. They were going to do this at some point. Rather than approaching criminal justice from that perspective, from that framework, rather you could say, listen, we just want to make sure that you don't do this anymore. You don't, inflict greater suffering on more people, right? And so, maybe their, imprisonment would be therefore justified. These types of things. Rehabilitation, of course, would be, obviously justified, and et cetera, right? And I think that is a more ethically justifiable framework for criminal justice, and it's more scientifically justified. So that I just sort of tied in so many different conversations into that one topic. But thanks. I mean, I love these topics. Thanks for bringing them up. Okay, low levels of vitamin B12 causes a lot of symptoms for me. One of them is forgetting and being unsure. Hamad, that's a group. You're absolutely right. Vitamin B12 is one of those vitamins that a deficit of vitamin B12 can have a bunch of pretty strange consequences, right? One of them can be confabulation, where people will lie, right, when you ask them a question, but they don't actually know that they're lying. They're just sort of making up answers, but they believe they're answers. This can emerge out of a condition called Korsakoff Syndrome, which can happen as a function of very, very severe alcohol ingestion. But of course, it can also, and I should say, part of the mechanism by which alcohol induces it is by reducing the absorption of a variety of vitamins and minerals, among them vitamin B12. But vitamin B12 deficiency can also cause like pins and needles sensations in your extremities. We know that this can happen for people who recreationally use nitrous oxide, laughing gas, the type of gas that they use in the dentist's office. Usually most people, when they consume that substance, it's a gas, right? That substance is, they do it, on a small scale. It seems to be largely harmless. Bad things can happen. But when not used too excessively, it does seem to be fairly harmless. That's why it's used medically. But it's when people start going way overboard, and they'll get like a big tank, like the size of a tank that they use to fill up balloons, like that type of thing, with nitrous oxide, and they just sort of like just sit there on it all the time. Obviously there can be oxygen deprivation if you're not getting enough oxygen, but beyond that, just a high consumption of nitrous oxide can be associated with vitamin B12 deficiency, which then causes that pins and needles sensation, numbness. Interestingly, however, once vitamin B12 levels are replenished, that sensation goes away. Pretty cool. All right, on YouTube, youtube.com/anthropoid. By the way, I just talked about how it is that we come to trust each other, or more accurately, what happens in our brain and in our body when we decide to trust somebody, which is kind of a funny way to frame it after going on and on about how free will, it doesn't exist. What is it? What do I even mean by decide? But youtube.com/anthropoid. There's a link in my profile page on Periscope. Also, check out haps.tv, HAPPS.tv. Link in my profile as well. It's a great new live streaming platform, great community. All the scientists with whom you might be familiar are over there now, so go check it out. DC Hooper, cosmologist, studies dark matter. Isa Betancourt, entomologist, studies bugs, has access to wonderful collections. Christian Zasa, wonderful nature photographer, astrophotographer, PhD in physics or in optics. And I think that those are the main ones. We're trying to get some more too. Let us know if you're interested in talking about science. Okay, so back to YouTube. I wonder how trust is affected by cognitive visualization in the person. Visualize as familiar, stranger, negative, positive. What is a stronger influence, sight or mind? What if born blind? Yeah, super interesting topic, Frank. I had the same thought, which would be like, this would be interesting to. So I talked about this study where this group at Leiden University in the Netherlands measured brain activity during, or not brain activity, but physiological responses when people are playing a game called the Prisoner's Dilemma. I have a suspicion that Justin might be quite familiar with the Prisoner's Dilemma, but it's a game of trust. And the nugget of the experiment was people were conducting this experiment where you decide to trust someone or you decide to betray them. Either they could see each other's faces, no talking, just facial expression, or they were looking at a blank wall. And so, it would be super interesting to see what happens in somebody who's born blind, who's never had never had the type of visual experiences that you and I have. However, most of the time when people are born blind, they have an occipital lobe, right? They have a visual cortex. It's not plugged in, right? People can be born, congenitally blind through a variety of mechanisms, but it's just that the rest of the visual system isn't, intact. So, but they still have all those neurons. The visual cortex is gigantic. We devote a lot of nervous system real estate to the brain, to the brain. Yeah, no kidding. You don't need a PhD in neuroscience for that one, but a lot of real estate in the brain to visual perception. And so there's a lot of neurons in somebody who's born blind, just kind of sitting there, capable of being activated. And so blind people can have visual experiences when they dream, and so on. But so, basically what we were talking about in this study is that a lot of these responses are hardwired, right? They're basically like, they're functional almost, they're functional at the age of 7 months old, right? And I don't know if you've ever seen a 7-month-old baby, but they're not particularly, they're not going to go out and hunt, right? They're pretty helpless, and perhaps even earlier than that. And so, how much of this is hardwired? And somebody who's blind, do they have a lot of the same responses despite the fact that they're not seeing facial expressions? That would be very interesting, because if they do, then that would suggest that at least the visual facial perception isn't necessarily central to all of the things that we talked about on YouTube. However, if they don't have those reactions, then we know. So yeah, that would be a really interesting study, man. And so and then visualizing someone as trustworthy or adversarial or something. Certainly, I mean, I think that would play a huge role. Another way to probe this whole system would be spritzing a little bit of oxytocin in the noses of people who are playing this game. And we already know that if you give someone oxytocin, which is a hormone that's involved in a wide variety of things, but particularly social cooperation, it's very critical for that. It's also involved in like maternal attachment to her, infant, lactation. It's involved in a lot of things. But it would be interesting to know if these physiological measures basically the fundamental argument was that in addition to cooperating, people who cooperated tended to physiologically synchronize. So certain measurements of their physiology tended to be the same when they were cooperating, when they trusted each other. So it'd be interesting to note if you give a group oxytocin, they will almost certainly trust each other more to a higher degree than a group that didn't get oxytocin. But then do all those other physiological measures also synchronize? They measured a bunch of things in this study. Specifically, it was skin conductance that synchronized, which is like how sweaty, how much blood flow is going through different parts of your skin that changes moment to moment as a function of your sort of inner experience. It'd be interesting to see if oxytocin cranks up that synchrony, where now you get, pupil dilation synchronizing, even eye movement synchronizing, maybe even heart rate synchronizing. That would be interesting. And of course, so how would that happen? What would that effect be, right? It would be in exactly what we're talking about. It would be in, in sort of nudging how it is that somebody's perceiving another person, and facial perception will be a huge part of that. So yeah, how to maintain B12 high? Well, I mean, so generally speaking, if you have like a reasonably competent diet, you should be getting enough B12. But it is one of those vitamins that it's pretty harmless to consume, and so if you're really concerned about it, you can just get vitamin B12 supplements. They even make like vitamin B12 gummies. Right. But, I suspect if you are attributing some kind of source of suffering in your life to a B12 deficiency, I suspect it's probably not the most critical culprit in whatever it is that's causing your suffering, unless you've gotten like a blood test from a physician and they've measured, circulating levels of B12 and they're like, wow, you have unusually low levels. That's okay. Great. Problem solved. Just go get some vitamins. But generally speaking, when people are suffering for some reason, when they're, they find it difficult to sleep, they are depressed, or their memory is beginning to fail, right? All of these things. Generally speaking, when there's a tendency, I think, for people to attribute all, those sources of just low, reduced quality of life to a specific vitamin or specific mineral or specific neurotransmitter. And that's just not how our biology functions. It's never just one thing. I mean, well, it's extraordinarily rarely just one thing. So is there an objective way to determine whether someone has appropriate levels of neurotransmitters? Oh, I love that topic. Oh, so there's so much to that. And I love that you're even thinking about this. So first of all, well, okay. Part of the reason I love this is that I did this for one of the main experiments of my dissertation. I measured changes in concentrations of serotonin specifically in a specific part of the brain. Well, the ventral hippocampus. You've probably heard of the hippocampus. So, first of all, is there a way to measure levels of neurotransmitters? Yes, absolutely. There's actually a few ways. The two main methods would be a technique called microdialysis, which is, you might have heard of dialysis. Same thing. You're basically just using a filter to filter out certain things. When people go on dialysis because of, an organ failure, they're basically compensating for the insufficient filtration of blood by going on dialysis, right? We're just using the same concept. It's just the filter is much, much smaller. It only enables certain, molecules to get through. And then there's also in vivo cyclic voltammetry. Okay, these are very difficult techniques, and to be clear, they're never done in humans. So they're done in animal models. They're done in, they can be done in primates. They could be done in humans, but to what end? And so here's the more important part of the topic you just brought up. Is there a way to determine whether or not somebody has appropriate levels of neurotransmitters? Now, I do not blame you for perceiving consciousness as a byproduct of perhaps optimal levels of certain neurotransmitters, right? You need to have somewhere within this window a level of dopamine, a level of norepinephrine, of serotonin, of glutamate, GABA, blah, 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 all these neurotransmitters. There's an optimal window that's generalizable, that applies to everybody. And that's just actually not how it works. What is a rewarding level of neurotransmitter, level of dopamine for you in one part of the brain? First of all, if you were to increase, let's say I artificially crank up dopamine levels in a part of your brain called the nucleus accumbens, the ventral striatum. We do this all the time. If you ever drink coffee, if you ever smoke cigarettes, drink alcohol, you do amphetamines, opioids, you're artificially doing that. You're cranking up that dopamine level, right? That'll probably, you'll like that. You'll enjoy that. Animals will self-stimulate. If you put an electrode down there, they will just sit there and stimulate until they can't anymore. At the expense of food, they'll choose stimulating that over eating food, that kind of thing. But if I were to crank up dopamine levels in a different part of the brain, you would find it extraordinarily unpleasant, right? And so all this to say, there's not like a universal level of a given neurotransmitter that is optimal throughout the entire brain. It's all about where those neurotransmitters are within specific circuits of the brain. So, what is rewarding in one part of the brain is aversive in another part of the brain. Okay. Another thing is that, human brains vary quite a bit. Now, there's a lot, most of the things about the human brain are similar, right? We all have a cerebellum. We all have a temporal lobe. We all have a frontal lobe, etc. We all have, anatomical structures. You have a hippocampus. You have a set of raphe nuclei. You have a ventral striatum, a nucleus accumbens, etc. An amygdala, a pineal gland, etc. Right? We all have those structures, but the interesting point is at a more granular level, which is we all vary at a more sort of microscopic level where, okay, yes, you have a ventral striatum, a nucleus accumbens. Well, let's do something. You have a hippocampus and you have an amygdala, right? Sure. But, the degree of connectivity between your hippocampus and your amygdala may be completely different from mine, right? Or the degree to which your amygdala connects with the rest of the brain may be much lower than mine, in which case I might be more predisposed to experiencing fear given the same stimulus that you would find maybe just sort of irksome, but not fearful. Okay, so we vary quite a bit on that level. We vary, of course, on our neurotransmitter receptors, right? Where you might have a form of the serotonin receptor that is let's say more common among humans. I might have a slightly quirky version of the serotonin receptor that has a slightly different amino acid. Receptors are just chains of molecules called amino acids, big molecules, that are then fold up into complex shapes that we call, in this case, a receptor. So I might have a slightly different amino acid at a specific location relative to you, and so our serotonin receptors behave very, very similarly. But we know that if I have that unique form of that serotonin receptor, I will be predisposed to developing depression given some kind of traumatic experience, whereas you probably won't, right? Just because of that one slight variation in our genes. So all this to say, the level of inter-individual variation is pretty substantial. I mean, this is true. Certainly, as far as I'm aware, you and I are not related, right? If we were related, let's say you were my sibling, then our brains would very likely, very certainly, be more similar than to somebody relative to somebody to whom we're unrelated, right? Let's say we are fraternal twins, right? Dizygotic twins. That means we share 50% of our genes, right? Or at least, right? And, fraternal twins are more genetically similar than just normal siblings, right? Then our brains would be even more similar, right? Let's say we're identical twins, right? And that means our genomes are the same. And hell, let's say, we are monochorionic identical twins. Meaning, not only do we share all of the same genes, but we grew up in the same placenta, right? We're about as identical as a human can be. Everything was the same in the womb. Well, not everything. That's the thing. So, even among identical twins, there will be variability in things like synaptic connectivity. Over time, quite likely, white matter volume, gray matter volume in different regions, just because we're not literally experiencing the same life. We're not in the same body, right? I'm exposed to more sun because I tend to sit on the side of the car that we both drive, to work or whatever. And so, I get more sun exposure than you get. And as a result, that's, whatever. As a result, I have different vitamin D levels or, whatever. I develop some kind of a tumor or something like that. So all of this to say, when you say, like, is there a way to measure whether or not somebody has sufficient or, let's say, optimal levels of neurotransmitters? The answer is yes, but the more important answer is that there's really no such thing as the one size fits all concentration of serotonin that we all need to live a happy life, right? So that doesn't exist. And so what I would suggest to you And I should say, like, you're not like, you're not crazy for thinking about it this way, because for the longest time, neuroscience and neuropharmacology, psychiatry, were sort of in this framework of seeing consciousness and specifically psychiatric conditions as a byproduct of chemical imbalances, right? I'm sure you've heard that term. People still use that term. And, it's not completely wrong, or it's not completely wrong, but it's also not completely right. Psychiatric conditions are not just a byproduct of, let's say, chemical imbalances of too little serotonin, too little dopamine. Because if they were, we would have cured all these conditions by now. They actually emerge from specific patterns of circuit activity, of neurons that are communicating with other neurons in a specific way that therefore generate depression, or therefore generate euphoria, right? Or resiliency against depression. And of course, neurotransmitters play a very important role in determining those patterns of activity, but it's not the end all be all. And so that's why it's so difficult to treat psychiatric conditions, because all we have are tools, pretty crude tools, by the way, to change levels of neurotransmitters. We don't really have, except for some very specific exceptions, we don't really have any tools to really interrogate or interact with circuits on their level, right? Literally modify circuit activity. Now, in some very select cases we do, like, for example, deep brain stimulation for the treatment of depression or addiction, but that is certainly not the norm, right? That is a very, very seldom applied technique where you're literally injecting current into the brain, into a specific part of the brain. So I know that was a long tangent, but and I love that you're thinking about it that way. There are people that don't absorb vitamin D and other things. Yeah, totally. Yeah, I mean, certainly there are studies, Betty, I'm sure you've seen them, that show that, generally speaking, adults, not just in the United States, tend to have suboptimal levels of vitamin D. That's a thing for sure. Yeah. Cool. Dark matter. Yeah. You should check her out. Can you explain what opiates do to the brain? Sure. By art, not cocaine. Why not by both? So sure. So opiates, the term opiate refers to molecules that are derived from the opium poppy. Okay. So morphine, for example, codeine, derived from the opium poppy. Opioid is a broader term that incorporates opiates, right? But it also includes synthetic or semi-synthetic opioid molecules and what they share in common is that they all interact with a specific receptor in the brain called the mu opioid receptor. Mu, like the little weird U character, but you can just type mu opioid receptor. And so that is part of our opioid signaling system in the human body, named because obviously it interacts with opiates, right? And so the mu opioid receptor is a very versatile receptor. It regulates a variety of things, among them, perhaps most intuitively, pain. So the mu opioid receptor can inhibit certain forms of pain, not all forms of pain, unfortunately, which renders it a suboptimal tool for the treatment of a lot of forms of chronic pain. But unfortunately, it is also one of the only things we have that's even has a modicum of efficacy. But that's part of the reason for why physicians are a little bit more reticent or apprehensive to prescribe opioids now, given all the overdoses and stuff like that. So opioids, let's just focus on oxycodone. Okay, oxycodone is an opioid. It's derived from what's called a thebaine alkaloid from the opium poppy, but it is you don't extract oxycodone from opium poppies. Anyway, so you take some oxycodone, let's say in a capsule, in a pill. And it takes a while to get through your stomach until it hits your intestines, and then that's where the oxycodone is actually absorbed. And so then it's absorbed, it gets into your bloodstream, your circulatory system, of course, and then ultimately winds up in your brain, where it will bind mu opioid receptors. And as I said, mu opioid receptors can inhibit the perception of certain forms of pain, but both fortunately and unfortunately, it also regulates affect. It also regulates mood and euphoria, but it's not like the kind of mood that, like cocaine, for example, to take an example from your handle. Cocaine almost directly just cranks up dopamine and serotonin levels. Or oxycodone does that too. But because the mu-opioid receptor regulates more than just, pain and mood, it regulates things that are like deep sources of like gratification and satisfaction, right? So, there's a ton of opioid receptors, for example, mu-opioid receptors in a part of the brain called the periaqueductal gray, which is a region of the brain that it looks like a donut, actually. And it encircles the cerebral aqueduct, if you were curious. Anyways, so there's a ton of mu-opioid receptors in the PAG, the periaqueductal gray. That region of the brain is also known to be associated with maternal attachment, right? And so we're talking about things that deep. And so this same system, and it sort of makes sense. Oh, and I actually talked about this on HAPS, actually, the relationship between mood and pain, emotion and pain, how they may very well be inextricably linked. That, pain only exists because we have an emotional reaction to it. And so it makes sense that this system would regulate both pain and mood, right? And so, this drug is binding this mu-opioid receptor, it's a complicated receptor. It does a bunch of things happen in the neuron once these receptors are bound. But broadly speaking, there's two main pathways. Ultimately, what happens is, the activities of certain neurons are inhibited, and that causes a reduction in the perception of certain forms of pain, an increase in affect, right? Or let's say an improvement in affect. It makes you feel good, right? And if it's just that only one time, you're one and done, you go to sleep, you wake up, whatever, there's no big deal. The problem is that when opioids are chronically administered, when people are regularly using oxycodone or whatever, heroin, the problem is that this receptor, these drugs, interact with this receptor in such a way as to cause an adaptation, right? So that there will be fewer mu-opioid receptors available. Those mu-opioid receptors will be less sensitive to binding endogenous opioids, and on, right? It's essentially all of these adaptations occurring to make you less sensitive to opioids, right? That's how you develop tolerance. That's how you develop a substance dependence. And in some cases, that's how you develop addiction. And so, interestingly, there's a really cool effort going on in medicinal chemistry to try and develop opioids that interact with the receptor in a slightly different way. In a, what is it called? It's called like an active selection way or something like that, where because of the way the drug binds the receptor, you don't get what's activation of something called beta-arrestin. Don't worry about that. But basically, it's just like it reduces the propensity of the brain to develop a tolerance to opioids, essentially. So that effort is going on. But fundamentally, that was your crash course in opioids. I could go on. There's more to talk about. But LSD versus alcohol, sure. So these are very different drugs. Alcohol is kind of a different drug relative to all other drugs. It's because it's such a simple molecule, and it interacts with probably every neurotransmitter system you can name. Gosh, something on my eye, and I cannot get it. Sorry. It interacts with obviously GABA receptors. That's the sort of primary receptor system with which it interacts, or at least it's described as interacting. But it interacts with NMDA receptors, glutamate receptors, in other words. It interacts with serotonin receptors, with mu-opioid receptors. If you block mu-opioid receptors when somebody's drinking alcohol, they will not continue drinking. They will not self-administer alcohol, which is part of the reason for why treatments like Suboxone are being explored for the treatment of alcoholism, because the opioid signaling system is critical to alcohol, euphoria from alcohol. And so, Suboxone has an opioid in it called buprenorphine, as well as another opioid that is actually an antagonist called naltrexone, it's naloxone. It's either naltrexone or naloxone. They're both very similar drugs. But basically, it's sort of like activating the receptor and then blocking the receptor at the same time. It's like a very controlled way to engage the receptor, as opposed to like something like methadone that just like activates the receptor and really stays on that receptor for a very long time, for such a long time, which makes it both useful and also difficult to withdraw from. But anyways, so alcohol has what you might call a dirty pharmacology or promiscuous pharmacology. It even interacts with ion channels. It does things like changes the membrane fluidity of neurons. It makes neurons like it changes how rigid the outer membranes of neurons are. It does like a bunch of wild things. It changes the concentration of salts in your like semicircular canal and in your your otolith organ, basically your vestibular organs, which is at least part of the reason. It's not the main reason, but part of the reason that you get a little dizzy when you drink, right? Or a little woozy. It does all of these. It's very, very promiscuous. LSD is far less promiscuous, and it's a much bigger drug. LSD is like a weirdly large molecule, and it primarily interacts with pretty much one neurotransmitter signaling system, but a bunch of parts of the signaling system called the serotonin signaling system, the serotonergic signaling system. In particular, it binds the specific receptor. There's a ton of different receptors, something like 14. I think there are more serotonin receptors than any other type of receptor in the body, more different variants, right? So LSD primarily interacts with one, 5-HT2A, though it does interact with other ones like 5-HT2C. But anyways, and because of where those receptors are in the brain, you get and LSD also will influence how serotonin itself is trafficked in and out of synapses. And so it's just sort of like it is sort of cranking up the volume on serotonin signaling, but because of where those receptors are, you get a bunch of really interesting effects, right? The psychedelic hallucinogenic effects, where your sensory perception obviously is altered. So you're not necessarily typically actually like it's not a delirium. Like, you're not seeing things that are completely not there. You're not just totally fabricating people that aren't there and stuff like that, which I talked about a plant earlier today on YouTube, youtube.com/anthropoid. The drug in that plant, a very common plant, it's probably in your neighborhood if you live somewhere in the South, is a deliriant, which is kind of crazy that it's so common. But anyways, and by that I mean you will have like full-on complete hallucinations. I actually have a crazy story involving that. Not the plant, but a similar medication that I misdosed one day. Maybe I'll tell it. Maybe I'll tell it on Haps. Anyways, so but because of where those 5-HT2A receptors are, you get altered sensory perception. You get altered mood, of course, altered cognition. There are moments of synesthesia, which is where sensory modalities are crossed, where you start like hearing colors or seeing sounds. But perhaps most commonly, it's you get these sort of like, you can get these kind of like tactile reactions too, or the sense of touch to seeing things, right? That's synesthetic. And there's a bunch of other interesting effects. Generally speaking, LSD is not particularly toxic, right? It doesn't kill neurons. Alcohol can. LSD is not addictive in the way that alcohol is. Alcohol is obviously quite addictive. Though people, that is not to say that LSD is a completely benign substance, right? Bad things can happen if you take LSD. If you take LSD one time, bad things can potentially happen, much less likely than other drugs, but potentially if you're predisposed to schizophrenia or mood-associated psychiatric illnesses, LSD can exacerbate those. Same goes for alcohol, by the way. So like, it's not like this is just unique to LSD. And people can develop compulsive usage patterns, but it's not an addiction in the same sense as like alcohol or cocaine or nicotine. And so yeah, I've talked about stories of like people who have accidentally taken like gargantuan doses of LSD. And so that's another difference. Alcohol, the dose of alcohol, obviously most people dose it in fluid measurements, right? Like mils or milliliters, or maybe fluid ounces, or it wouldn't be fluid ounces, probably shot glasses, ounces, I guess. But you can measure it in grams too. Whereas LSD is administered in micrograms. So that is such a minuscule amount, that is so low that like basically if you can see the LSD in powdered form, you're looking at hundreds of thousands of doses. Whereas alcohol, if you can see it, you're looking at maybe just one drink, right? Yeah, I mean, there's other differences. I could go on, but those are the main differences. Ovaltine. I'm not sure what you mean by that. As a neurologist, what do you think of an Alabama hot pocket? I don't know what that is. I'm not a neurologist. I'm a neuroscientist. But I don't know what that is. How about DNA? Is it the same? I'm not sure what you mean by the DNA. Sorry about that. Yeah, sorry about that. Well, the DNA, I'm trying to remember back to what you might be referencing. Oh, the DNA, maybe for the identical twins. Yes, yeah. But although their epigenome will likely have some nuances. So, you have your genome composed of DNA,
A, T, C's, and G's, wrapped up in these chromosomes, they just sort of crammed together, spun together. But then, essentially on top of your genome is an epigenome, epi on top of your genome. And these are different markers, different molecules, literally molecules that are attached to your genome that will determine whether or not, or when, how much, and where a given gene will be expressed. And so that you don't inherit, you can inherit that from your parents, but it's subject to change, whereas your genome does not change over your lifetime. And generally speaking, if it does, if one cell has some kind of a genetic change because you've been bombarded with a bunch of, ultraviolet radiation from the sun, or you've been smoking or something like that, then that can cause, aberrant cellular activity, right? So, but that because the genome has changed, whereas your epigenome does change. More recent evidence suggests that not only does your epigenome change, and so that sort of contradicts the kind of central dogma of genetics, which is, DNA to RNA to amino acids to protein, to expression, and the DNA is immutable; it does not change, it vindicates at least a little bit an old scientist named Lamarck. Was it Anton Lamarck, I think, who basically this was when we still didn't really know about natural selection as a mechanism of evolution. We knew evolution was a thing, we just didn't know how it happened until Darwin and Alfred Wallace discovered it, explained it. One of the competing theories was Lamarcanism, which is like, you pass down, we knew that traits are heritable, and so his argument was that you pass down acquired traits. You lose a hand, your child is more likely to not have a hand, right? That kind of thing. Obviously not true. However, there's now evidence that epigenetic changes can be passed down transgenerationally. Pretty crazy, right? So Lamarck wasn't 100% wrong. Pretty cool. How do you change your brain chemistry? Well, brookslove.com, you have no choice. You're changing it right now. By even having that thought, you have changed your brain chemistry. Your brain chemistry is changing literally millisecond to millisecond. Now, I know I'm being sort of like irritating there. What I suspect what you mean is how do we intentionally change our neurochemistry to achieve a desired effect, right? Like how can I change my neurochemistry so that I'm not depressed, or so that I'm smarter, or my memory is better? It's going to vary completely, right? Let's say that my goal is to just increase, oxytocin levels, increase serotonin levels. Then I would probably go snuggle with my daughter or with my wife, right? Cuddling, that'll just crank up those hedonic neurotransmitters. If I wanted to crank up some dopamine levels, I would, drink some coffee, drink some tea, drink some beer or something. That's how you do that. But really, I think when people are understandably and frankly intuitively focused on neurochemistry, when they're trying to optimize some aspect of their lives, I suspect that you're not going to get the biggest bang for your attention there, because, again, like neurochemistry, it's very, very important, but it's not the main event of consciousness. Right? Consciousness is a byproduct of specific neurons communicating with other neurons in specific ways, specific frequencies, like how many times is this neuron in this brain region sending signals to this neuron way over here, and how frequently, right? And that is influenced by neurotransmitters, but it is not exclusively influenced by neurotransmitters. And so really what if you're like, let's say you're interested in improving your memory or getting better at the guitar or, something like that, or just, becoming more informed about a given topic, don't worry about the neurotransmitters. They'll do what they need to do. What you need to focus on is practice, right? It's the practice that is the nutrition. It's, it's not necessarily like your levels of dopamine. So, but, we all do this, not all of us, obviously, but many, many, many humans do this every day when you drink your first cup of coffee, right? You're artificially altering your neurochemistry to achieve a desired effect, which is increased energy, increased motivation and focus, and perhaps a reduced deficit as a function of insufficient sleep, right? Because I think a lot of us get insufficient sleep. How can we restructure our neural pathways? Same type of thing, right? Like that, your brain is constantly changing. I mean, if you were just sitting in a room just a white room, white walls, silent, right? And you were sitting in, white clothing, whatever, and you had like no stimulation, your brain would still change in response to that. And so you change, like in a sense, things like psychotherapy, the whole point, the whole goal of psychotherapy is to change neuronal pathways, right? In a way that is therapeutic, right? And so it is the practicing, it is the learning, right, that is responsible for that you're interested in achieving, right, but is associated with changes in neuronal pathways. Like when you learn how to play the guitar, your brain has physically, literally changed. And so, but the goal wasn't to change your brain. Your goal was to learn the guitar, right? This is not a vaccine. No, I don't have a vaccine, but I cannot wait until I do. We almost got an appointment today. That's close. There's a school that had extras laying around, but anyways. Is DNA the same as well in identical twins? Yes, right. I'm addicted to ibuprofen, Tylenol, and Benadryl. Ibuprofen and Tylenol. Well, you can't get addicted to ibuprofen and Tylenol. You can't get addicted, right? I know what you mean, but like, you can be dependent, right? Although even still, these drugs are just incapable of inducing the kinds of effects that addictive drugs induce. But you can become dependent upon their effects. Now Benadryl, that's different. Benadryl, I suspect, can have an addictive component to it. What's interesting is that diphenhydramine, or is it diphenhydronate? Diphenhydronate, I believe, is what's in Benadryl. It's essentially the same molecule as diphenhydramine, but those molecules, those antihistamines, that are also used, as perhaps you're well aware, as sleep aids over the counter. They were part of the initial set of discoveries that led to the development of Prozac, of antidepressants, because these drugs, these antihistamines, have some serotonergic effect. And it turned out there was the observation that people who were suffering from severe depression, and keep in mind, this predates antidepressants, right? So there wasn't really any serious treatment apart from pretty, intense ones. They just recognized that when administered this antihistamine, the symptoms of their depression would abate. They would be relieved of their depression. And it was like, well, what is going on with this? This antihistamine? What's the deal? And that very soon thereafter emerged the serotonin hypothesis of depression, which, as I think is very clear from our conversation so far, is not entirely true, right? The serotonin hypothesis of depression. Serotonin obviously plays a very important role in depression, but it is not the sole determining factor of depression. What's up, the notorious Triple X? I wish I was as smart as you. You are, Michael Gorilla. Well, if you're a gorilla, then perhaps you have a point. But no, you are. Listen, I am no smarter than anybody here. I am like 100% I could not be more confident. Like, I've had a lot of years to prove it to me, right? That I am no smarter than anybody in here. It's just that this is my job to understand these topics, right? I'm a neuroscientist. If I didn't know about the brain, I'd be a pretty crappy neuroscientist, right? And there's nothing magical about this information. You can definitely learn it. It's just a factor of how much time you spend learning it, right? That's it, I assure you. How about sugar versus alcohol? Awesome topic. Yeah, I mean, well, so there will be some similarities between sugar and any addictive drug, right? Rewarding drug. But there are more differences than similarities. The main similarity is that, the only reason that people consume these substances, these rewarding, recreational substances, is because they have some effect fundamentally on dopamine levels. They have a bunch of other effects that are also very important, but every single drug that somebody does voluntarily, in one way or another, will crank up dopamine levels in a circuit in the brain called the Mesolimbic dopaminergic circuit. Also the mesocortical limbic dopaminergic circuit, right? It's a broader circuit, but fundamentally it's dopamine levels that are coming from basically like right where the brainstem inserts into the cerebrum, sends dopamine via these pretty long projections to a part of the brain called the ventral striatum, the nucleus accumbens. And sugar also, when consumed, will increase those dopamine levels in the same regions. However, not nearly as much as any addictive drug. And you don't develop the same kind of desensitization or sensitization. Cocaine, for example, induces dopamine receptor sensitization as opposed to desensitization. Both of those are sort of two sides of the same coin, where receptors can either become less sensitive to alcohol or to heroin or whatever, or they can become more sensitive, right? Where it's like, they have an even greater response than they did before consuming the drug. That's the case with cocaine. Kind of crazy, right? Anyways, so, sugar doesn't necessarily do that. It's certainly not to the same extent. So there are, I think, some really meaningful differences, but there are also similarities. I mean, and of course there are similarities, right? Like anything you do, any decision you make to consume a given food, to talk to a person at a bar or something, to study for a test, to prepare for an interview, all of those things emerge from activity in that same circuit, right? That motivation to engage in these behaviors emerges from activity in that mesocortical limbic dopaminergic circuit. And so it's no wonder that a food that cranks up activity in those circuits, you're going to want to eat that food. It's going to feel good. It's going to feel rewarding. A molecule that happens to crank up those dopamine levels, same story, right? It's because it is the circuit that is devoted to determining your motivation to engage in any given behavior. So yeah. Sometimes I suddenly forget what I was going to say, and I freeze and shy. Why is that happening? Call me Botan. I think we've spoken before, have we not? Anyways, yeah. Well, that is that's a natural thing that happens, for everybody. Our memories are not perfect. Our working memory is not perfect. So it's natural for that to happen every now and then. If it's happening quite regularly, then you should definitely see a physician. That could be a sign of, some potentially concerning conditions. And when you get shy, I mean, yeah, so you have this whole emotional reaction to something that is fundamentally a cognitive event, right? The cognitive event is I'm trying to express a thought, trying to verbalize a thought, right? That's purely cognitive. That does not involve mood. But everything involves mood. Everything you do, every moment you're alive involves mood in some way, shape, or in some form or another, right? And so this is a negative thing. You don't want this to be happening. And you now recognize that this has happened to you more than once. And so you have an immediate aversive reaction. And my bet, total speculation, but my bet is that reaction, that aversive reaction, exacerbates the problem. Because now you're not focused on the sentence, you're not focused on the thought, on the memory, now you're focused on the fact that you forgot. And so you're devoting cognitive attention to something that has nothing to do with accomplishing your goal of articulating this thought. And so if I were in your shoes, it's happened to me too, of course. If I were in your shoes, what I would do is just recognize that it happens to everybody, and there's no reason to be shy about it, no reason to be embarrassed. Just be like, brain fart, oh well, next. And it'll come to you right when you stop talking to this person, right? As it always does. Oh, of course, that's what I meant to say. Yeah, but sincerely, like if you are experiencing this on a regular basis, that could be a sign of something that you would want to take care of. Ian, what is the best place to go after Periscope? Best place to go for me, two places. You can see them in my profile. YouTube.com/anthropoid, YouTube, where I just talked about what happens in our bodies and in our brains when we decide to trust somebody. And then the other is a live stream. It almost just happened to me, Botan. I almost just forgot what I was saying. But the other place is a new live streaming platform called haps.tv. So pretty much, we've managed to get pretty much all the scientists that were regularly using the platform to all, join up on Haps. And, it's free. It's like any other streaming platform, but better community. Founders are really competent, and I think they have a good philosophy, good approach. They have backgrounds in journalism as opposed to backgrounds in like Silicon Valley startup culture and stuff like that. And just a great community, a lot of tools, a lot of great production tools that can be used. And so, yeah, I know the platform is going to grow. So, now is the time to jump on board. So I have links to both of those in my profile on Periscope. Thank you for asking. Your hair is fantastic. Yeah, not necessarily. I've only had one haircut throughout this whole ordeal, right? COVID-19. My wife cut my hair. Funny story. What happened was she was using normal scissors, and she's great. She has a background in science, you know, she's very detail oriented, taking her time, taking a very long time, and cutting my hair. And then I got, you know, this cheapy, you know, set of scissors on Amazon that has one of those scissors with the teeth on it, you know, they're like little gaps to thin out the hair, I think. And so she was like, you know, we were both like, oh, this is going so great, like let's get crazy, like come on, let's use this thinning shear thing. And so, you know, we're just jiving. I think I was playing some music or something, and then she puts down the normal scissors, does something I can't remember, and then picks up the thinning or intends to go pick up the thinning shears, but she picks up the normal scissors and just goes and immediately goes, oh no. And so she took this like huge chunk of hair off this side of my head, and so I immediately went from having just a trim to fundamentally a new hairstyle. So that's pretty funny, but I appreciate the sentiment very much. What percentage of the brain is fat? You know, I've wondered that. That's a really cool. So I can't tell you for sure. I can't tell you what percentage, but I can tell you that a hell of a lot is. So, and here's how we know. So first of all, if you look at a slice of human brain, you will see primarily two things: white matter and gray matter. And generally you'll see a lot of white matter, right? And so what white matter is, it's all fat. I mean, not all, but it is white because it's fat. It's myelin, right? These are, you know, connections between neurons that are literally wrapped around in fat as an insulator. And so there's a lot of fat just there, but then every single neuron itself is surrounded in fat. Its membranes are, they're phospholipid bilayers, so lipid, right? Lipid is a fat. And so every single neuron has fat in it. Not everyone, but most neurotransmitters are released in what are called vesicles. Vesicles also have lipid layers, right? So they're like little water balloons made out of fat. On the inside is where the neurotransmitters are, but the sort of outer shell is all fat. What else? There's, you know, astrocytes. Those are you know, astrocytes, glial cells, microglia. Those also have a bunch of, you know, fatty membranes. There are, you know, cholesterol. You know, you synthesize cholesterol in your brain, use it to make a bunch of different hormones, testosterone, estrogen, and that of course has fat or is associated with fat. So it would be a lot, like a pretty substantial chunk. You know, but I couldn't tell you. I couldn't tell you like what the ratio would be. I would just be wildly speculating. Maybe over 50%, you know, maybe something like 65% maybe. But seriously, like I can't even think of how I would try and quantify that. Okay, warning, clumsy question ahead. What are you driven to want, like more, to satisfy your scientific curiosity or personality? The empirical evidence or the plausible fantastic theory combination? It's the story for me. Yeah, no, I totally get what you're saying. There are definitely scientists, neuroscientists, who are really mostly driven by just, you know, figuring out how an ion channel functions. You know, they are content studying the muscles of crabs for decades, right? And it's not because, and maybe they just don't really talk about it, and it is more romantic in their mind that it is contributing a thread to the sort of tapestry of our understanding of human consciousness. But for me, that's just not enough. Like I am definitely driven. I was, you know, brought to science through science fiction, and so it's the stories for me that are the most interesting. Okay, does science need our bodies after we don't? Yes. Yes. All right, I should probably call it a day. So thank you, everybody, for hanging out. My name is Ian McLaughlin, in case you are unfamiliar with me. I have a YouTube channel called youtube.com/anthropoid. Link to that in my profile page. Today I talked about what happens in your brain and in your body when you trust somebody. And then I'm on a new streaming platform called Haps, haps.tv, HAPPS.tv. New live streaming platform is where it seems like everybody's going to go after Periscope is shuttered. And so I have a link to that in my profile here on Periscope as well. Other than that, I will see you one of the last few weeks that we have together on this platform. Next Sunday. So I hope you have have a happy St. Patrick's Day if you celebrate that. Stay home and don't go to bars and stuff. But or if you do, wear masks and socially distance. Otherwise, I will see you next Sunday. Have a great week, everybody. Stay safe. See you there next Sunday. See you there. All right, thanks everybody on YouTube for hanging out. I appreciate that. Again, any input you have, please let me know. I would love, to be clear, there's certainly things I would like to do that I just don't have the time for at this point. Like I'd like to cut these sections up a little bit more. But other than that, any ideas for topics, I would love to hear it. If you haven't subscribed, I would appreciate that. If you would, if you find this interesting or you want to, show me how I'm wrong about dualism. And if you haven't liked, maybe like. Otherwise, I will see you next time. See you, everybody.